the coordinator of 2 ERC projects. He received several awards, including the Young Investigator Awards from the Louis Janté Foundation and in 2015, and he was also elected a NEMBO member in 2016. This year, he was recognized with the Excellence in Scientific Research Award by the University of Porto, and he founded the Y Science, a pilot project that brings together scientists to promote science education and the scientific method in young children. So Elder, thank you for joining us.
Seja as pilhas. Estará a reconhecer ali qualquer coisa? Já não está a reconhecer aqui. Anyway. Uso este. Ok. Perfeito. Obrigado. Ok. Okay, I think we're on. Thank you very much to the organizers for picking me to give an overview of our use of the core facilities and how this is used to learn about a specific problem that we're interested in, which relates to the problem of cell division. And I chose this title because I think it goes both ways. So it can go from technology to new knowledge, but also new knowledge can drive the development of new technology. And this is what I would like to cover today in this short presentation. Cell division, I'm not going to tell you everything that we know about cell division, but I'll give you an overview of the process and the problem. Essentially, you will know that without cell division, we would not be here as multicellular organisms. We need cell division to undergo the complete embryonic development to become multicellular organisms. Just to give you an idea, in our body every day there's about 1 trillion cell divisions going on. So it's kind of an important process, not only during embryonic development, but even in the human adult. So we need cell division, for example, just to keep a good immune response, for example, or to allow regeneration of tissues, and also to allow sexual reproduction. Very importantly, and if you do the maths, so with all these cells dividing every day, we know, based on in vitro studies, that we live under the constant threat of losing or gaining particular chromosomes. So every 200 divisions, there's the potential to lose or gain one particular chromosome. And again, if you do the maths, this kind of brings the point, or makes the point very strongly, and I cannot avoid mentioning that the gain or loss of particular chromosomes is just the most common feature of all human cancers that we know. So it might not be the origin, so it's quite controversial in the field, but it's the most conserved and the most horizontal feature that is common to all human cancers. And to understand the basis of cell division and how chromosome segregation is accurately occurring, we need to understand the process called mitosis, which was kind of highlighted in this movie. This basically is a 2D representation of this movie. I'm highlighting a particular structure here that I'm not going to detail. It's called the kinetochore. It simply forms the interface between the chromosome and the chromosome segregation machine that is mediated by microtubules here in green. So kinetochores will be in red, microtubules will be in green. So then you can sort of see how this entire choreography from the very early onset of mitosis to the separation of the sister chromatids at the end of mitosis, you need to go from a rather chaotic or sort of distribution of each individual chromosome until they organize at the equator of the cell in a process called chromosome congression, and then each sister chromatid separates to end up in two daughter cells that are genetically identical to the very original progenitor. And my lab is particularly interested in understanding how this whole choreography is coordinated both in time but also in space. I'm not going to get into too much detail, but I want to give some highlights of how we have been using the core facilities at i3S to understand more about this process. And I'll start with one that is, of course, very dear to us, the light microscopy. We are hardcore microscopists. For example, by using the technique called fluorescence recovery after photobleaching, we can express fluorescent molecules with a fluorescent protein, and by photobleaching with a specific laser with a specific wavelength, we can determine, for example, the half-time of a particular molecule in a particular structure, and also inquire whether this molecule has a stable pool and a mobile pool. So it's a very simple technique and gives us actually a lot of information at the very molecular level about the dynamics of almost individual molecule dynamics in a particular location. And I highlight this work because it was the very first paper that we published as a lab in 2006, and was very much due to the use of this technique in the core facility. We also use the animal house, this is just to highlight so the protein that I showed you before, so we can really go from the molecule to understanding the physiological relevance of the molecule for the organism, because we can generate knockout mice for this particular molecule. This molecule is called CLASP. It doesn't matter for now, but gives you a clear example. For example, when you compare a wild-type animal with a CLASP1 knockout, you can see that this knockout is clearly much smaller than the wild type, and actually it's overall smaller, but if you focus on particular morphological features, for example, the brain is clearly smaller, an indication of microcephaly, which is a common feature of some of these proteins, but allowed us to investigate in depth the physiological consequences of the loss of this molecule. We also use the flow cytometry unity quite a lot because we deal with a lot of stable lines expressing fluorescent molecules. We like to establish stable lines stably expressing these molecules. And just another example of a recent work, and these are all lines that were somehow selected using this laser device that sorts out the fluorescent cells that we want, so we can deplete endogenous protein and then express different mutants on particular functional domains of this protein. I will not get into detail, but it will teach us how specific molecular features translate into function and relevance for mitosis. And just to give you an example here, so this is how the wild type looks like. So the different colors actually represent different fluorescent proteins or fluorescent tags. And these are some mutants expressed in a knockdown background against the endogenous protein. You can see some missegregation events suggesting that some of these functional domains indeed are required to ensure mitotic fidelity. These are just other examples. I will not lose much time with it. More recently, we have benefited quite significantly from the use of biosciences screening facility to rather than focusing on a particular molecule, provide a comprehensive overview of a particular process. And this process is the challenge that we're looking here is the alignment of the chromosomes at the equator. So more than 100 proteins have been implicated in this process, but most of these studies were based in fixed cell material. So they just took snapshots, and this doesn't tell you about the dynamics of the process and which aspects the particular molecule accounts for. And so we decided to perform this systematic molecular dissection of chromosome alignment in human cells. And with this, so we took more than 100 different siRNAs against potential candidates. We did live cell imaging for all of these 100 different candidates, image analysis, and then sorted the phenotypes into different functional groups. And I highlight here 4 of these functional groups. Phenotype 1 is basically no phenotype, so it's normal. Phenotype 2, there is a delay in the process, but they ultimately make it. Phenotype 3 is that they never make it, so they cannot complete the process. Phenotype 4, they complete the process, but then they fail to keep the chromosomes aligned. So all these 4 categories indicate, so if you take a snapshot, it will tell you exactly the same thing, but they actually mean completely different. The underlying reasons are completely different. And again, just to give you examples, I hope the contrast is good enough. So this is no phenotype, so normal cell, no problem in congression. They go from chaos to order, and then the sisters separate, end up into daughter cells. Phenotype 2, you go and you end up with a few chromosomes that are misaligned, but after a delay, they ultimately make it to the equator and then they divide happily. Phenotype 3, they completely fail congression. So from the very starting point, there's always a few chromosomes that are unable to segregate, and because they are unable to segregate this cell, after a lot of time, they have to do something. And one thing that they do quite often is to die, and it's actually thought to be the basis of many chemotherapy agents used in cancer treatment. Phenotype 4, completely different, so they actually make it to the equator, but they cannot keep it there. And if you just compare, for example, this stage here with this stage here by a snapshot, you'll think, oh, it's exactly the same thing. But the history tells you that it's not the same thing. So these proteins act at different levels. And this allowed us to classify these in different functional groups. Again, going back to the molecule, so the B2 Tech facility, which is more on the biochemical and biophysical analysis of proteins, was very helpful very recently to understand the biochemical nature of this protein clasp, and indicated us that it forms monomers in solution, both using recombinant protein, but also the endogenous protein in cells. We can also not only investigate these features by combining size exclusion chromatography with centrifugation gradients, but we can also use more sophisticated techniques such as dynamic light scattering, which basically measures the scattered light that these molecules emit due to their Brownian motion. And basically, it gives you an idea. So this single peak indicates that this population is homogeneous. So there's a single type, single population, so it exists predominantly as a monomer, we think. And if you do this as a function of concentration of the protein in solution, if you see that, in fact, the diffusion coefficient decays with concentration, it actually tells you that although it's a monomer, it tends to self-associate under particular concentration. This was kind of important for us at that time because we did not know, and the literature was quite controversial, and we have to thank these people also for their great help in understanding this. Then we also highlight some of the contributions of the histology and electron microscopy facility, again looking at the physiology of the depletion of these proteins. So these are actually lung sections of wild-type mice. These are alveoli. You can see that these spaces means that they're actually full of air. So these lungs are fully functional. If you look at the class 1 knockout mice, I forgot to mention that they actually die immediately at birth because they cannot breathe. They cannot inflate their lungs, and this is proof of it. So there's no air in the alveolar, and if you just do these simple tests of putting a lung floating on a buffer. So a wild type lung will float because there's full of air. The class 1 lung just sinks straight to the bottom because there's no air. And these are just histological proof for what we were able to see both at the functional level in the animal, but also using these complementary tests. And now, so sometimes it's very important that an institute like i3S has very, very strong core facilities. This is key to keep cutting edge research and keep the competition at the highest level. And sometimes core facilities, they kind of average. They average good because they are good pretty much to everything, but sometimes they are not exceptionally good for particular aspects that deals with your research. Sometimes we need to give this extra edge, and sometimes based on what we know, we develop technology that allows us to give this extra kick that we need to understand what we're seeing. So the reverse. So how does knowledge allows the development of technology? And here I would like to highlight 3 technologies that were developed or implemented in my lab. And although they were developed by my lab and they are managed by my lab, I'd like to highlight that they are open to the entire i3S and outside community. So whoever wants to use it, feel free to contact us. So this technique relies on laser microsurgery in living cells. So basically we use laser light to go deep into a living cell and manipulate single structures. And I'll give you some examples. So we can use laser light to cut through a chromosome arm, for example. And this is just an example. So we cut a small piece of the arm and the arm immediately moves away. And sometimes if we do the same experiment, the arm will move in a completely opposite direction. This, I will not get into detail, but just this simple experiment told us that these motors that exist at the kinetochore interface between the chromosome and the microtubule are the key elements of the chromosome that teaches the chromosomes where to go. If you rely on motors sitting on the chromosome arms, it can go anywhere, either to the right place or to the wrong place. As you see here, the chromosome will ended up expelled towards the cortex. The other technique is basically an inducible way of generating fluorescent speckles on structures. And this is a fluorescent spindle, so it expresses GFP tubulin, and with a simple pulse of light that creates this like a light rain of so it's just simply speckles of light going through a diffuser element in the light beam that ends up eating the specimen. You'll see that suddenly the spindle becomes speckled, okay? And we can use this, I'll tell you exactly how do we use this, but we can do this in different stages of mitosis. If we do it slightly later when the microtubules are more stable, you can see that the speckles last longer, even by just simply visually inspecting these cells. But also you can photoconvert different fluorophores. So in this case, the tubulin is green, and we're going to photoconvert into red speckles. So you get a red and green pattern that gives you information. For example, if you look at an individual speckle, by measuring the motion of the speckle over time, you can determine a property known as microtubule flux in these cells, but also by measuring the loss of fluorescence, you can infer about the turnover and half-life of a particular structure within these megastructures called the mitotic spindle. By the way, there's many Pereiras, they're not the same person, it's a very common Portuguese name. And very lastly, probably you have heard of super resolution microscopy techniques. One of them is called STED. It's very powerful because it's like a confocal microscope, but it gives you this extra kick in resolution. So it's about a 4 or 5-fold gain relative to the diffraction limit of light. So you can get resolutions up to 50, 60 nanometers with a light microscope. But yet this was not enough for our purpose because our object is rather messy, is rather complex. And I'll give you one example where António Pereira in our lab developed a slight modification that improves this technique for our particular object of interest. And this is what we get with conventional STED. So you can see that you get pretty good resolution at the individual microtubules when they are separated enough. But if you look at this bulk here, it's a very complex structure, very noisy. We cannot resolve the elements in this very complex 3D environment. And by simply playing, so this is called a phase mask. It's used in this technique. I will not get into details, but what he did was to generate a bivortex that is completely out of phase. So you have an internal vortex that is one phase, and then you have an antiphase, small ring vortex. And by just simply combining this phase mask, you now compare the bottom part of the same object. You can see now, so you actually lose a bit of the resolution on the individual microtubules, but you start resolving structures within this messy environment. And here in more colorful view of the same problem, so you can start detecting individual microtubules forming this structure, whereas here you cannot really resolve them. Same if you compare the green and red. This is just a very side-by-side comparison. For example, at the exact same laser power, which in a conventional STED microscopy actually gives you great resolution on single microtubules, for example, it's not going to help you in a complex environment. At the exact same laser power, this is the kind of information that we can retrieve. So I think even any layperson can blindly compare these and say, well, this is much better, and this is what we were able to achieve for our particular purposes. And with this, I'd like to thank many people in my lab that over 3 generations already contributed to the work that we do, and not only using and taking advantage of the existing core facilities, but also developing technology that we feed back to our research to learn more and gain new knowledge. Many collaborators, both internal and external, the core facilities and their heads for their amazing help, and these agencies for funding, and I'll be happy to take any questions. So thank you. Any questions about cell division, about the use of facilities, about the new technology that is available to all of you? No. Paula, get a glass of wine.
Thank you, Elder. Very nice, as usual. My question is the following. Do you feel that here at i3S you have any technical constraints in terms of support, in terms of the platform? So it's not people, of course, it's the... if you feel that somewhere else, you will have more possibility to develop your work.
Thank you for the question. It's a very interesting question. So I set my lab here in Porto 15 years ago, and for the entire 15 years I never felt limited in what we wanted to do for a single second. So I think so whenever we feel limited, so we arregaçamos as mangas and we develop what we need. Of course, you need the right people, you need the right drive. So I never felt limited. Of course, for specific aspects, again, so a microscope in a core facility has to satisfy 100 different clients with many different purposes. We have specific purposes. So sometimes what we have available is enough, sometimes it's not enough. And whenever possible, we develop that. And whenever we cannot develop, so we collaborate. But I think this is true anywhere in the world. So no one has at their disposal everything. It's so much better to live here than anywhere else. So, in short, we don't feel limited. Of course, by increasing in size, I think one very important aspect to take into account is not only diversity in terms of experimental approaches, but also taking into account the scaling aspect that one has to fit to satisfy the needs of 200, 300 customers rather than 50 as it was early on. So just having the technology is not enough. We need to replicate sometimes the technology that is heavily used. And of course, this is an investment. So the better technology, the better access that we have to the technology, the better work that we will produce. And at the end, this will feed back to the success of the institution, I think. After all, there's questions.
Can I ask a question? Sure. Elder, in your lab, people have to deal with very fundamental aspects of science, but they also have to deal with new technologies. Do you feel that they are prepared when they come to your lab to deal with these technologies? If not, what do they have to do?
Well, they love it. It's like a toy to a child. It's a new toy. You can have everything established that they know. If you give them a new toy, they all want the new toy. It's just human, I guess. So the learning curve is not that steep. It's very gradual, and over time, we are admittedly a microscopy lab. So maybe for some other labs, you need to understand how to purify protein and analyze protein like best thing in your life. But for us, it's microscopy. So they learn it, they become proficient, and then they can teach others and take this knowledge with them wherever they go when they leave my lab. So I think it has to be challenging as well for young people. If you give them something that's trivial, it lacks this pinch of salt that we all want to have when we do science. It's this little extra edge that will lead us to a new frontier of knowledge that I think people appreciate.
So thank you, Elder, for sharing all of those examples.
It's just a tiny bit, trust me.
I understand. So, but regarding these interactions, you used a lot of different facilities with different questions. And I'm thinking about the same question using different facilities, and I'm thinking of correlative light microscopy. Have you thought about that?
We would love to do that. And I think we do need to invest in things like, I've been talking with Rui for, maybe more than 10 years. We need to develop correlative light electron microscopy as a standard approach. This is the gold standard internationally, but to my knowledge is done well by a handful of labs, if not less. So it takes the effort to learn, but then once you know how to do it, it's really... we are now back, electron microscopy is now seeing a new age of relevance, because it was critical when it was developed to see at the ultrastructural level what we could not see anywhere else, any other way. And nowadays, with the development of fluorescent microscopy, of course, it gives you this dynamic edge that tells you the history of or the relevance of a particular molecule to a process. But then we still need to go deeper and understand, okay, where exactly is this thing going? So having the capacity on the same cell to tell, okay, we have the history, then we fix, and then we look, oh, it's right here. It's critical. I would love to do that. I think we're paying a bit the price of the dimension. Rui and his collaborators, maybe I don't think they can handle the demand that they have at the moment to be able to invest in developing this new edge. I would love to do that. We have been doing it in a collaboration elsewhere in my previous lab, because I know that they do it well, and they don't have the demand that we have. So, but I'm sure they're willing to teach. If you want to put this effort, yeah.
Elder, thank you so much for showing how you can use the potential of our platforms in a very elegant way, and also to provide these new infrastructures that will be available to our researchers. And I was going to ask you, how could you use them, for instance, in particularly the STED that you have demonstrated? Is it possible to easily adapt it, also to visualize all those cytoskeleton interfaces, but with
Very
cell
easy.
matrix interactions?
Absolutely, it's trivial. The only condition that you need to do when you do STED is to take into account the fluorophores that you use, because everything is in the far red region, and it's a less conventional region to when we do fluorescent microscopy. So for STED, you need to use far red dyes or far red proteins or far red conjugated secondary antibodies. So this is the only thing that you need to take into account, and these are well established now, and we have them. So you just need, instead of using a conventional secondary antibody with Alexa for 536, I don't know, you just use these other secondary antibodies with the far red shifted dyes that will be appropriate for STED. You also need to, because you're looking at with higher resolution, you have to increase the concentration of your antibody so that you fully decorate your structure. So because of the diffraction limit of light, in a conventional light microscopy, a dot will look like a huge balloon. So we will not be able to see these differences when you use diluted dyes. Here you will see them. So you have to concentrate so that you decorate uniformly the structure that you're interested in. And in fact, many people in the institute have been using already the facility. I'm thinking Monica Souza, I'm thinking Didier Caban, I'm thinking Sandra Souza, and probably many others that I can't remember now.
Thank you very much.
You just have to come to us and find a spot. Okay, I'm done.
Okay, so our next speaker is Maria José Oliveira. She's a graduate in biology by the Faculty of Sciences from the University of Porto, and she holds a PhD in medical sciences by the Faculty of Medicine from the University of Ghent in Belgium. Her postdoc training was dedicated to the role of E. cadherin and Helicobacter pylori on gastric cancer, and she initiated at INEB a new line of research focused on the role of human cells on the modulation of cancer progression. She is a principal investigator at the i3S and also an invited associated professor at the Institute of Biomedical Sciences Abel Salazar. Her work is focused on elucidating the role of tumor microenvironment on the modulation of human cells and on the promotion of cancer cell invasion and immune escape, dissecting the underlying molecular mechanisms and developing immunomodulatory therapeutic strategies to counteract cancer progression. Her work was published under over 70 papers in international peer-reviewed journals. She also received the L'Oréal First Woman in Science in 2009 and the Yamajiwa Yoshida Memorial Cancer Study Grant in 2016. So, Maria, if you would like to join us, thank you.
Thank you very much, Sonia.
You're welcome.
So let's try. Yes, this. Okay, so thank you so much. First of all, I would like to acknowledge the organizers for the invitation to give the opportunity to present our work here. And I will structure in a way that I will try to show you our scientific questions and how, in fact, the scientific platforms have been fundamental on addressing all these questions that I wanted to show. I try to illustrate a little bit of the story of our research in the last 5 years and try to demonstrate what we have been mainly focused and how you have been so important important for us at the scientific platforms. I just wanted to tell you that some months ago we had here an inviter, a collaborator from Gustave Roussy Institute, and it was very happy, not that we didn't know of that, but it was very happy to hear from him in the first of his talk, saying that he was impressed with the structures of the Institute, particularly with the scientific platforms, and showing how luxury we were that just going down one stage, we could have all these facilities available with human knowledge. And for us was important also, of course, coming from abroad as an important compliment to you. But I wanted to say that I think the scientific platforms, as Professor Mario already illustrated how rich and important they are, how still we need to improve, and we can improve that, and there's emerging infrastructures also that Elder made available, show that we have a very important human knowledge, and I think this is the important sell-up of our platform. And I found across our investigation a lot of enthusiasm coming from these people and completely dedication. So, and I wanted to show what they have been contributed to. So, in fact, we are in our group dedicated to tumor microenvironment. And what is this? This means that when we look to a tumor, the tumor is not only formed by cancer cells. And besides the cancer cells, we have many cellular components derived from bone marrow-derived cells, but also endothelial cells, fibroblasts, neurons, adipocytes, and immune cells that share the same compartment with the tumor cells. But we have also a very important cellular component here illustrated, that is the extracellular matrix. Altogether, they comprise the tumor microenvironment, and it's very important because these cellular and the cellular components regulate the activity of the cancer cells, but they are also regulated by the cancer cells themselves. And from our work, we have been centered particularly on the role of an immune cell population that are macrophages. And although we have been now working also with the T regulatory cells and dendritic cells, part of our study has been made in this population of cells, particularly because these cells seem to play a different role in terms of tumor progression, because monocytes might differentiate up in certain environment in pro-inflammatory, producing pro-inflammatory cytokines that recruit at the site a couple of immune cells like CD4 T cells, CD8 T cells, NK and NKT dendritic cells that mount an effective immune response against the tumor, promoting tumor elimination. But we know that this is not always the case. That's why cancer arises. And because there are also other elements, particularly enrichment at the tumor microenvironment as immune suppressive molecules that turn these macrophages anti-inflammatory, and therefore they produce anti-inflammatory cytokines. They maintain the immune suppressive environment, which is not only impairing the elimination of the tumor, but at the same time are producing molecules that promote the invasion, the angiogenesis, and the metastasis of these cancer cells. And that's here where we have been working. So we have been trying to understand how macrophages can regulate cancer cell activities, but how cancer cells, and particularly the extracellular matrix, can modulate macrophage polarization, but also try to understand how this crosstalk goes in the presence of radiotherapy that is frequently used to treat rectal cancers, and at the same time to understand how these mechanisms might be established that we can develop more efficient therapeutic targets to modulate macrophage and cancer cell crosstalk also under hypoxia conditions. So therefore, we have been using and making part of all your platforms in a one way or another that have been keen in helping us to address all these questions, and I will try to illustrate that. So at first, we have tried to characterize these populations of macrophages, and we have made a detailed characterization in terms of electron microscopy of these M1 and M2 macrophages, and we have, using the histology and electron microscopy platform, but also advanced light microscopy and in vivo chem assays to achieve the data that we demonstrate here. So we could demonstrate that, in fact, these pro-inflammatory macrophages are less efficient in stimulating cancer cell invasion in comparison to the with the anti-inflammatory macrophages, immune suppressive. They are less efficient in inducing the migration of cancer cells, in sustaining their proteolytic activity, and also less efficient in sustaining their ability to induce new blood vessel formation. So the anti-inflammatory macrophages seems in vitro to be the ones that, in fact, as it has been described in breast, to counteract the immune surveillance and at the same time sustain the invasion and the activities associated of cancer cells. But we wanted to know what happened, in fact, in human samples, and therefore we needed to characterize the histological sections of cancer patients. And we had performed that in a straight collaboration with histology and electron microscopy platform, would help us also to mount this system to identify some of the markers that we can now use as markers of pro-inflammatory and anti-inflammatory macrophages. And through this long-through characterization, we could visualize that the macrophages are distributed along not only the tumor, the invasive front, but also at the normal adjacent mucosa. But that what we could find is that particularly at this invasive front, we have a shift of the macrophage populations, with the pro-inflammatory macrophages reducing over the anti-inflammatory macrophages. So this is confirming our in vitro data that, in fact, the macrophages of anti-inflammatory activities are the ones that might sustain these activities towards invasion and towards invasion ability, and that's the one we can, in fact, found at this intratumor region and at this invasive front. We wanted then to understand how important the extracellular matrix could be to regulate macrophage function. And to do that, we have assessed, through based also on histology and electron microscopy data platform help, we could assess to human samples that were directly obtained for surgical resections. And we wanted to take fragments of the normal, but also of the tumor, and we desolarized to obtain the extracellular matrices. These matrices were then desolarized. We could confirm that desolarization retained the major ECM components and also maintained the structure of electron microscopy, as you can observe and appreciate here. But we could use these matrices then to repopulate with monocytes. These monocytes differentiated as macrophages deep in the matrices. And what we could observe with the collaboration with genomics platform was that indeed the macrophages that are now differentiating over the tumor matrices are expressing a completely gene card comparing with ones with the normal matrices, being the fact that ones on the tumor start to lose markers of pro-inflammatory activity and they gain the expression of anti-inflammatory activity. So this corroborates our data from in vitro and also in vivo data, showing that as at the invasive front where we find these anti-inflammatory macrophages, that indeed the tumor matrix might have a role on that, driving these macrophages, that they are human macrophages, into an immune suppressive phenotype. We are currently, with the proteomics platform, trying to detailedly detect what are the enrichment factors that are present in the tumor and absent in the normal matrix, not only of ECM components, but also soluble factors that might be aprisionate, to try to understand how these might drive cellular differentiation into a different profile. We then tried to understand how these crosstalks could be involved in the terms of response to radiotherapy, and here the bioscience screening help was really fundamental. We were visualizing, we wanted to irradiate macrophages because we know that when we have a tumor, not only cancer cells are irradiated, but also the macrophages are irradiated, and we don't know how this irradiation might affect the activity of the macrophages themselves. And we visualized that all the irradiation, in contrast to the tumor cells, the macrophages were not dying. And so it was very important to demonstrate whether there was DNA damage in response to radiation. And there it was with the help of the bioscience screening platform that we could not only quantify the phosphorylation that was occurring by macrophage, but looking on each macrophage and quantify the fluorochrome intensity that we could have inside of each cell. And this helps us to demonstrate that, in fact, after one cycle or even 5 cycles of irradiation, there was an enhancement of histone phosphorylation, indicating of DNA damage. But as I told you, these macrophages are not dying. They enhance survival pathways, and they are still sustaining invasion, but also they are sustaining the angiogenesis of cancer cells, as it was demonstrated in the corallantoid membrane angiogenic assay. We also performed the proteomic analysis on these irradiated versus non-irradiated macrophages to try to understand what are the main differences in terms of protein expression. And we could observe that there were several proteins that are different in the irradiated versus the non-irradiated. And this gel-based and gel-filler-free approach led us to find of some molecules, in particularly cathepsin D and transferrin receptors, that are systematically altered in the irradiated macrophages. So we see that there is a decrease of cathepsin D and a consistent increase in transferrin receptor, and we are now running a project based on these results, trying to understand the importance of iron metabolism on macrophages and in response to radiation. We also, based on the genomics, evaluate how these radiation turned these macrophages, and we could observe that the macrophages that were irradiated were losing anti-inflammatory markers, and they were performing here some enhancement of pro-inflammatory markers, in particularly co-stimulatory molecules, the CD80 and CD86, and also of MHC class II molecules. So this is showing that macrophages, although present in the tumor with immunosuppressive activity, with irradiation they can become into a pro-inflammatory activity, but they still sustain cancer cell functions. And that was important then to lead us to the last question I wanted to show, that was to the development of immunotherapeutic approaches trying to counteract what might occur at the tumor site. And as you know that we have there immune suppressive cells, the idea was to develop nanoparticles that could counteract this immune suppressive environment, turning these cells more pro-inflammatory. And we did that in collaboration with the group of Professor Mário Barbosa, taking advantage from their knowledge of these molecules that were known to be pro-inflammatory. And these nanoparticles were extensively characterized, and through the biointerface nanotechnology and also bioimaging platforms, we could identify the particular size of these nanoparticles, also their charge, showing that they have a positive charge, what is important for the engulfment into cells. And we demonstrated these nanoparticles were internalized through confocal microscopy in the macrophages, but also on dendritic cells. Through the help of the cytometry platform, translational cytometry platform, we could detail in, I think, very well what is the effect of these nanoparticles on immunosuppressive cells. And we could observe that while dendritic cells, in the presence of nanoparticles, start to express more presentation molecules, but also co-stimulatory molecules, releasing pro-inflammatory cytokines as IL-6, IL-12, and TNF, they are also inducing the proliferation of CD4 T cells. In contrary, in macrophages, the nanoparticles did not alter the presentation of antigen at their surface, but they decreased the number of scavenger molecules that are characterizing these anti-inflammatory macrophages, releasing pro-inflammatory cytokines, and on their turn, inducing the proliferation and activation of CD8 T cells. We demonstrate that in these macrophages, and these cancer cells were then able to counteract cancer cell invasion once cold cultured with the cancer cells. We then tried to know on the base that the radiotherapy seems to be very good because it seems to be promising and promoting the pro-inflammatory activities of these macrophages. But they could not be probably enough. And to avoid some recidives that might occur, relapses of the tumors, we decided to combine in an orthotopic model the nanoparticles with radiotherapy. And therefore, these experiments were not performed in our animal facility because we had limitations, because we needed these irradiation platforms. So we have performed these experiments in Ghent. But what we could affirm was using a mouse model where we introduced the mammary cells we have combined the treatment of radiotherapy with or not nanoparticles. And what we have in this data is that in fact they seem very promising because the nanoparticles, when combined with radiotherapy, seem to decrease the primary tumor growth. And we are currently analyzing here this data with the help of the histology and electron microscopy, but also the translational cytometry unit platform. And we are verifying the population and characterizing the myeloid and lymphoid populations. But we could also observe that these nanoparticles seem to be very promising because in combination with radiotherapy, they seem to decrease the size of metastases in lung. And instead of having big metastases, we start to have small metastases in the lung tissue. And this seems to be accompanied by an increase systemically of immunosuppressive molecules. To last, and this is my last slide, I wanted to show you that we are not only focused on cancer, but we are also looking on the models of diabetes too. And we are particularly using here in our animal facility a core of animals that have been using as models for diabetes. And this is important because, in fact, diabetic patients have normally impaired vasculogenesis that might lead to very severe effect that may lead even to members amputation. And we wanted to understand how relevant is the extracellular matrix important for the revascularization of these tissues with a kind of therapeutic possibility to improve this interface. And therefore, what we have done was using these mouse models that are also available at the Institute in case you might need for approach your questions. We have selected the different organs, we decellularized the organs, and we are now characterizing in detail the liver, the lung, the heart in terms of structure and histology. We are characterizing the matrices regarding their stiffness, and we already visualize that on the diabetic models, there is the stiffness of the matrix is increased in comparison with the wild-type animals. And we are now using these matrices to recolonize with endothelial cells to induce the vascularization that we might quantify and integrate this biological data into the theoretical model of angiogenesis. And we are doing this in collaboration with the physics department in the University of Coimbra. So I wanted to acknowledge the people that have contributed to this work, particularly the previous and members and the current members of my group here underline how our collaborators, some of them I already mentioned during the presentation, and of course the scientific platforms for your input, for your not only technical help, for running so well all these equipments, but especially to provide your scientific input. That's why what I get when I go downstreams and downstairs and I speak with you. So thank you very much. So if you have some questions, reasoning. Okay.
So I just, it's more of a question is to ask for one of your comments and to give a comment. First is what you said in the beginning, that we are so lucky to have all of these platforms, scientific platforms, and for those who have started 20 years ago in Campo Alegre should know that to have a genotype of a mice, we would receive a tail, and from that our owners would have to get to the results, and now we have it in our computer because there's a facility, two facilities doing this, and also sequencing. We started the sequencing. If we had money, we would send it to a company to do the sequencing, or if not, we would do it ourselves with big gels for Sanger sequence. This was 20 years ago at IBMC NEP, probably Epatimope would be the same. And now this saves us time and then allow us to make more complex questions because we have all of this in our hands. But so the question is, because I see you use transmission electron microscopy and scanning electron microscopy. And of course, we would love to have all of equipment in our institute. And Professor Mar was talking about how difficult it was to have small equipment, big equipment as well. But so I would like to know a comment, not specifically for electron microscopy, but when we want an equipment, we can either have a big project for equipment, or we can ask the institute to buy it, and usually we send the company here to make a demonstration, and then we see so I want to comment of your perspective as a PI, and how to deal with this need or this desire of having a specific equipment.
So this is maybe I make a first comment to the first part that you have made on how things have evolved, and in fact it's I can witness also that evolution, and of course many of the facilities we are starting asking abroad, some that we do not have here. But I think that in the last years we have been re-equipped with many equipment that when we compare the 10 or 15 years ago, that have been enrichment our knowledge. So I particularly focus on the new proteomic approaches that we have, the new flow cytometer, the cell sorter, so that have been keen not only for us, but for collaborations that we have. In particular, we have collaborations with colleagues from the University of Minho that are approaching the differentiation of macrophages along different tissues, and they are collaborating with us. Not only we are doing many experiments here, but we have the cell sorter facility that they do not have accessible over there. So I think we have improved a lot, and I think that of course we need to think that many of these equipments are being so well sustained because the scientific platforms are run by technicians responsible that made also at the same time the formation of these people that are daily working and assisting the work. But of course we need to think in the future, and we need to consider on these re-equipment possibilities that Professor Mario has demonstrated. And I think at the institutes we have to have both possibilities. We like Professor Mario also that showed that. I think we need to have in fact calls that are broad calls that are not dependent from the institute but are from the government, from the national level, that are applying for equipment calls where we can apply. And of course many of us have also applied to ERCs, as the case of Elder Nuno, that brought to here to the institute other equipment that we did not have without this funding. But at the same time, I think that we needed to have a kind of basis of the institute in order that we can reorganize in a certain way to have a special money back for this buying of some of these equipment, especially small equipment. As Professor Mario, I think this is very important. But as he mentioned also, it has been imputed at the institutes a huge amount, and understand, and many of us are appreciative of that, for contracts with human resources that are very important. So it's not easy for the institute, and I would say it's almost impossible to do better than it is in regarding of the buying of this equipment. What we have done punctually was in some institutes, we have many times some groups and group leaders we have joined part of our funding to buy together some equipments. And that's, for instance, we have bought a recent nanoidentator at the Institute to precisely approach so many of these questions that we needed and that we need to combine for biomechanical measurements. So this is a solution that we are also bringing together. Of course, this has limits, and has limits on the platforms, the funding that we have. And of course, I think we need to incite, as Professor Mario also mentioned, that given our quality, would it be possible, at least to the national level, to open also re-equipment fundings and calls where we could all together apply it, and even crossing with other institutions in big networks, as Rui also demonstrated, to bring back to our research updated technology and updated equipment. But of course, I think, and the Elder demonstrated that very well, that have been also a lot of individual application on this way, and that has enriched our institute. But the institute is also, as a whole, I think, dedicated to that. And as Elder mentioned, I'm very glad that I'm here. And I have been in Belgium before, and although it was a very good institute, we have all the things available, the sources were very much separated. It was not integrated. We have here a very nice conditions. Everything is integrated and well run. And more than over, you have scientific output from the people that are running the platforms. And I think that is one of the most precious part, is these human resources that we have.
Okay, thank you, Maria.
Okay, thank you very much.
We are closing this session now. We are going for a coffee break, and then we come back in half an hour for another invited speaker. Thank you. vacation mode after
Semi-vacation.
semi-vacation? Okay.
So I saw a tiny little bit of platform because my suitcase is in the platform.
It's always.
Or just the office.
There's water also here.
Yeah.
Yeah.
Very often.
Hello. So our next guest is Patrick England. Patrick England obtained his PhD in biochemistry and enzymology in 1992. After several years as a postdoc and researcher in protein engineering and structure function analysis, he decided in 2008 to dedicate himself full-time to the operation and management of the molecular scale biophysics core facility that he had created in 2002 in the Institut Pasteur in Paris, France. The facility has significantly grown over the years, and it has gained national and international recognition, first becoming a French national infrastructure in 2008, and more recently, through the launching and coordination of the Arbre Movio European Network, now a cost action, that brings together more than 150 molecular-scale biophysics facilities and resource labs from 30 European countries. Furthermore, he has been at the heart of the CTLS, Core Technologies for Life Sciences, Europe-wide initiative since its launching in 2010. He became the president of the CTLS Association at the beginning of 2018. Patrick will talk a little bit about the importance of networking with the core facilities in a global overview. So Patrick, please.
Okay. Hello, and thank you for the organizers of this open day for inviting me today, and it's a pleasure to be back here. I was here in i3S for the first time 2.5 years ago in another context. So in the context of the cost action, that is it important for core facilities to network, not only in their own field of specialty, but in transversal issues that are common for core facilities, whatever their field of specialty. And really, I have absolutely no intention to give lessons to anybody. That would be totally preposterous and stupid. But here, I would like to discuss with core facility staff, with core facility users, with administrators. And so don't hesitate to interrupt me if you have questions during my presentation. Well, of course, you can keep them for the end also, but if you have questions during the presentation, don't hesitate. So, as was said during the introduction, I am both leading a core facility and leading an association of core facilities in Europe that I'm going to tell you about. So first, I wanted to make just a few slides of introduction about why, and this has already been stressed both by the core facility leaders at the beginning of this day, and then by the two lovely presentations by core facility users. So why is it important in modern life science research to have core facilities? And really, to remain competitive, we all know that one of the most important things to address successfully challenging questions is to have timely and convenient access to technological and methodological resources. But even more importantly, to the expertise that goes with these methodologies. And so, nowadays, most institutions, research institutes, since the 80s and 90s, because before, well, the term of core facilities really appeared in the early 1980s. Now, in most countries, and well, mainly in Western Europe and North America, most research institutions have recognized that the best way to have technological and methodological resources available and open for a wide scientific community is through shared resource laboratories. And you can see from one institution to the other, the semantics that are used to designate such laboratories can vary a lot. So core facilities is a very common name, but also you can see technological platforms, expertise units, research infrastructures. Here in i3S, it's scientific platforms. So the imagination is quite wide at this level, but basically I think all these terminologies cover basically the same thing. And as was stressed by your director at the beginning of the day, so having core facilities is certainly for an institution a significant investment. But I believe, and many of us believe, that it really is a cost-effective investment. So I would just like to refer you to two very nice publications, which I think are interesting both for core facility people and for core facility users and for administrators. So one of them was published in 2016 in EMBO Reports by core facility coordinators of many institutions. in Germany, in Spain, in Germany again, in Switzerland, in Austria, in Belgium. All of these people are members of the CTLS association that I'm going to tell you afterwards. And so this paper in the Science and Society part of EMBO reports really explains, I think, very well why core facilities are a very important prerequisite for breakthroughs in life sciences. So this is in Europe. Another publication I want to show you comes from the United States. And so this was led by an association. So ABRF means Association for Biomolecular Resource Facilities. So it's a wide organization in the United States bringing together core facilities in basically all fields of life sciences. And they published a very nice case study based on one single institution, which is the Northwestern University near Chicago, showing how they built a sustainable portfolio of core facilities, what were the difficulties that they met, how they overcame them, and I think it's a very nice example and quite recent, so from 2018. So I want just to briefly show you what is the situation in the institution that I work in, which is Institut Pasteur. At the moment, because the things have changed, as in any institution, the models change with time, with the use. Currently, in Institut Pasteur, there are 20 core facilities, and the core facilities are under the supervision of a director that is called Director of Technology and Scientific Programs, and which is a kind of scientific supervisor of the core facilities, and there is also a head of operations for these core facilities. The core facilities are distributed in 2 centers. There used to be only one, but then they were separated because there was a recognition that animal resources have constraints that are relatively specific comparing to other kinds of technological resources. And so at the moment we have 2 centers, so one for animal resources and one for technological resources. And really I would like to insist that it's important in the terminology, it's Center for Resources and Research. So meaning methodological and technological research. So here are the different core facilities that there are in Institut Pasteur. So anyhow, the slides will be available for you, so I won't comment on all of them. So here is the platform that I am leading. So you can see, just in terms of semantics, you will see that even within one same institution, there can be different semantics. So in Institut Pasteur, there are platforms, and there are what is called UTECs, which means Unit for Technology and Service. So the limit is not very clear. The idea was that some platforms were carrying out more technological research or methodological research than others, and were termed units of technology and services, contrary to platforms. But really, honestly, the limit is not very clear, but we carry on having this kind of dual terminology inside the institute. So here are 9 platforms, here are other 8 ones, and so this is for the Center for Technological Resources and Research, and so then in the Center for Animal Resources and Research, there are 3 platforms, so a central animal facility. I would say something that is relatively specific of Institut Pasteur, well, although they are also in other institutes, as our institute is very much focused on infectious diseases, and many infectious diseases are carried by arthropods and insects, so we have a special animal facility for mosquitoes in Institut Pasteur, and a mouse genetics engineering platform. So really, well, I think all of you both working in core facilities and using core facilities, from what I heard this morning, you are all convinced that basically the core facilities are providing much more than services and access. They really contribute on the one side to the visibility and attractiveness of an institution, And this is really important when you try and attract students, graduate PhD students, or you want to attract principal investigators, junior principal investigation, or you want to attract external collaborations. Really, the existence of core facilities, and not only the existence, but I would say the top-level operation of core facilities are one of the main instruments for an institution to attract all these categories of people. So really, core facilities are, beyond what they do internally, are a very important institutional outreach and networking instrument. Oops, sorry. I didn't get. Okay, so also one of the things that they are very important about, and that maybe wasn't stressed this morning, is the essential role of core facilities in training and dissemination of knowledge. Really, well, you showed a few courses that were organized here, but I think really in terms of training, especially students or postdocs, and having them progress, core facilities are often in an institution the main tool for training the students. Also, they are an excellent entry point for the channeling of funding and grants, so both instrument-based or research-based, and we see, for example, in Institut Pasteur that I would say between 40 and 50% of the grants of the institution involve core facilities as partners. And really they are put forward to have these grants being successes. Also, one of the very important roles of core facilities is their role in technological watch. And I would say beyond what you can usually do by publication tracking and going to congresses. And I think this is really somewhere where it's very important that core facilities are networked with peers to really sense where things are going, to have prior knowledge of developments at a very early stage before things become commercial. And this is really one of the very important roles of core facilities for an institution, so that the institution really is competitive. And something I heard this morning, and I think is very important, it's, of course, it's good to have things in-house, but I think it's even more important for core facilities to be well connected, to know whom to relate with when there is something that cannot be done locally. So I was a bit surprised. Well, it's often the case, but the two PIs who talked in the second part of the morning said, well, when I cannot do something locally, I think about whom I can collaborate with. To a certain degree is good, but for me, I think this is really the role of a core facility. If a PI comes to a core facility and the core facility says, well, we don't have the most appropriate instrumentation to do it, or it goes beyond my knowledge, it's in fact for the core facility to know whom they can address the PI to, whether it's in the country or it's outside. And really, I think that's really one of the roles of the core facility, to be connected and to know where to go when there is something that cannot be done locally. Of course, how can we have core facilities playing fully their role? So I think that one of the things that is most important, and I will stress afterwards, is the aspect of human resources. Of course, core facilities must have cutting-edge technological or methodological resources, of course, and also one of the, I would say, key aspects is the question of funding, as you all know. In some institutions that have set up core facilities, they had the impression that basically the core facilities were going to self-sustain themselves. And I think now, I don't know any institution that goes on having that illusion that a core facility is going to self-sustain itself. having a good performing core facility needs institutional structural support at the level of premises, at the level of human resources, at the level of funding, and at best one can expect only a partial cost recovery of the cost of the core facility. So what are, in my view, the key aspects for the success of a course facility? The number one, and the number one by far, is the question of expert human resources. You can have very glossy instrumentation. If you don't have somebody that really knows how to bring this instrumentation to its very limits, it's totally useless to have a very bright and nice instrument. So then the second one, of course, are cutting edge and tailored technological and methodological resources. A very important aspect is, and I think this is what you are doing today with the U Open Day, is to be really open to make constantly known what is available, both in terms of resources at the level of technology and about projects, and so to communicate all the time, and also to have clear and efficient procedures for project management. When a user comes to a core facility, they must understand how their question is going to be addressed, what is the procedure, and really, I would say basically everybody has to be treated in the same way. When a core facility starts going wrong, it's very often because people have the impression that some people are being favored with respect to others, and I think that has to be avoided by all means. In terms of human resources, I really think, and this was also said this morning, that what people really expect to find in a core facility are people who are technological and methodological experts. And these people also must have some kind of communication skills. Working in a core facility is not the same as working in a research lab. You have to be open to plenty of different questions, and the most unexpected ones. And a core facility manager or a core facility scientist has to indeed be ready to tackle any kind of question. And I think that this is really more important than being proficient at the level of management. So this is repeating again. So what users really expect when they come to a core facility is to find people who will address their questions, and sometimes the questions can be sometimes they can be very precise, and people know very well what they are coming to fetch in a core facility. But I would say, for example, to take as an example my core facility, I think more than 50% of the questions we get are, I have this problem, basically, what can you do for me? And so I think that's really the role of a core facility, to understand really what the user wants, try and reformulate the question, and come out with potential solutions to address these questions. And one shouldn't expect that it's really the user that is going to know what instrument they want or what procedure they want to use. I think it's really for the core facility to propose to the user, well, we could try this. If this doesn't work, then maybe we're going to try this. And if I cannot do it myself, then maybe I know the lab that can help you. And so being a successful core scientist really means that people need to be solution-oriented. They need to be ready to try and understand problems and to propose both standard and customized workflows and experimental setups. And another aspect that is important is also the expertise in data analysis. I think we all are, I would say, frustrated and angered sometimes when we see publications in which people misinterpret or overinterpret data. And I think this is really something where core facilities play a major role in saying, well, with these results, this is how far you can go in terms of interpretation. This is what you can say. This is what you cannot say. And I think probably most of you as core facility managers and scientists are very often contacted by journals to be reviewers. This is one of the things that many of us in core facilities spend a lot of time doing, because journals know very well that the people who are able to judge whether a paper is going too far or not are in the core facilities. So, what does it take to be good for a core facility staff? So, as I put it here, working in a core facility is not something that comes just like that. It should be, or it is for many of us, a real choice. And choosing to go into a core facility is going into a specific career track that is parallel to that of a research scientist, of a principal investigator. And of course, going into a core facility doesn't mean that you're going to be there all your life. There are possibilities to jump to and fro, but it's really a turn in a career. And I think, well, most of you who work in the core facilities, so there are many different trajectories, but it's not something that is just chosen by chance. And if it is, then it really needs to think why you are working there. A very important role of core facility scientists is to act as, I would say, second mentors for students and postdocs. So what do I mean by that? Is that very often, and well, this has happened for me at least 10 times, we end up having some kind of co-tutorship or co-supervision of PhD students, where the principal investigator is in fact supervising the biological question and the main biological trends of the project, but who is actually kind of supervising and leading the student at the level of the methodological approaches that enable progress is very often people from core facilities. And I think this is something that is important to be stressed, and that requires, of course, that there is a strong connection between the PIs and the core facility people. Unfortunately, what we see in our institution, and I have seen in many institutions, is that sometimes the core facility staff only see the student and never get to meet the principal investigator. And I think this is something that is quite counterproductive. There needs to be a direct relation, and I would say a kind of peer-to-peer relation between the principal investigator and the core facility people, so that they work together to enable the progress of the scientific research. Of course, and this happens very often, and I don't know if it happens, how often it happens here in i3S, but we see many core facilities in which there are PhD students that are actually directly attached to the core facilities and supervised by the core facility scientists, mainly to carry out technological and methodological development projects. So just to summarize at the moment what I see as being the main challenges for core facilities. So in many institutions, unfortunately, core facilities are relatively ill-defined and poorly positioned. Definitely not the case here in i3S. I think that you really have a wonderful setup. But honestly, in many institutions, it's very difficult to know where exactly are the core facilities. Sometimes they are kind of embedded within research groups, and so they are not visible. People don't know how to access them, etc. And so this is really a problem often. And also basically in the research landscape in Europe, there is often problems of knowing exactly how to define core facilities and their role. One also big problem is that very often core facilities are too isolated, in my view. That so here you have the possibility to connect between core facilities because there is a good geographical situation, and most of the core facilities here I see belong to either national or European networks. It's this is far from being the case. If there is something that they cannot do, they don't know where to turn to solve the problems. And so just before going to really to telling you about CTLS, so some critical aspects that, in my view, remain to be improved is very often, and I think speaking to core facility managers here, it's also a problem here, that we have to fight and struggle all the time to so that the institutions recognize that core facilities are genuine scientific structures. So I see that here the core facilities are recognized as scientific platforms, which I think says well what is the role of the platforms. A constant problem is to recognize, to have recognized what is the added value of the core facilities in terms of research, innovation, and training. And a recurrent problem is the question of how to see the contribution of core facilities recognized and acknowledged in publications, in grant applications. And so, well, if you have questions about this later, this is something that we can discuss. And also, in many institutions, the status of the people working in core facilities is not very clear. And so this is really something important, to have a clear status recognized for core scientists, for core technicians, for core managers. So where does CTLS stand for all this? Why was CTLS created? So really it was to address the challenges I've just talked about, and many others. CTLS, so which stands for Core Technologies for Life Sciences, was an initiative that was started informally between, I would say, a bunch of maybe 10 people at the beginning in 2010. And so our idea was that core facilities are sometimes and often well networked with other labs or other core facilities in their own field. So proteomics core facilities have the occasion to meet other proteomics core facilities, and etc. But that there wasn't really a place in Europe where core facilities from all different fields could meet together and discuss about things that were common to all fields of specialties. And so CTLS is really a bottom-up, community-driven and grassroots professional organization. So it's a kind of society. And in this society, we really recognize the diversity of the core facility ecosystem because, as I say, when you go from one institution to the other, you get very, very different setups. And I don't believe that there is one model that works best than all others. Models are very context dependent, and so we have to take into account this biodiversity as being enriching and source of inspiration. So starting in a very informal way, in 2016, CTLS actually became a legal entity. So it's an association which has its headquarters in Paris. And really, well, our motto is really by core facility people, for core facility people. This is really our objective, to bring scientists, technical and administrative staff that work either in core facilities or in association with core facilities. And as I say, whether they are called core facilities, research infrastructure, shared resource laboratories, whatever name, this doesn't matter at all. Also, there are many different sizes of core facilities or research infrastructures, from very local labs that sometimes are run just by one person to big disseminated research infrastructures involving sometimes tens of nodes. So it's just a question of size, but basically these structures do the same kind of job. And so the ambition of CTLS is really to cover all aspects of this ecosystem. So I won't go in detail through all this, but so on the one side, we want to provide to core facility people a hub where they can meet their peers. And also a role of CTLS is a role of advocacy, both towards decision makers and funders, to have them really realize, so both at national and European level, so that they realize what is the crucial role and the added value of core facilities in research, and also for the scientific community at large to have them understand what is the impact of core facilities and the importance of core facilities. An aspect that is important is the question of professional development. So it's important that core staff at all stages of their working life, from young trainees to senior personnel, have ideas about what progress they can have, what skills they can train, and this is an important role of CTLS. So, what is CTLS at the moment? Currently, CTLS as an association has 200 members, plus or minus 5, I would say, from 20 countries of Europe and beyond. So we are not strictly limited to Europe. We have a few members from North America, Australia, and Japan. But of course, the focus, and I would say natural focus of CTLS is Europe. As you see, Portugal is here. And so currently, among the 200 members of CTLS, there are 6 members from Portugal, but they all happen to be working in Lisbon. So they come from Instituto Gulbenkian de Ciencia and from Champalimau Foundation, these 6 members. And of course, people from i3S are more than welcome to join. So the membership of CTLS, as I would say any professional society, is on an individual basis. So currently the membership is 100 euros per year, and well, people can become member for 2 years. So we really try and incentivize people who are not core facility managers to join too. So there is a significantly reduced fee for people who are not core facility managers, who are technical staff or who are students that are working either in core facilities or with core facilities. And it's possible for an institution. We have several institutions that have decided to purchase a group membership package for the people working in their core facilities. And so there is a preferential rate in that case. What are the benefits of membership? So people who are members of CTLS can get very significant discounts on registration fees for our congresses. I'll tell you just afterwards what our congresses are and other events. They can also get bursaries to participate in these events. And we are also putting together a scheme of mobility bursaries to allow people from core facilities to go and stay for relatively short missions in another core facility in another country or in their own country, but preferentially in another country to have the occasion to acquire new skills. So this is biomedicine, and also people who are not scientists or technicians but are really administrators, and I think this is also something that is very important to address. So what is the current structure of the association? As many associations, the decision-making body is an executive committee. From the executive committee emanates a board of directors. So as was said at the beginning, I currently am the president of the association. But you see, well, the board of directors is from many different places. The vice president works in Sweden, the secretary in Germany, the vice secretary in the USA. So it's very multinational at this level too. And we currently have managed to recruit an administrative director, part-time administrative director, and an assistant that is also helping us for administrative tasks. I would say most of the work is carried by working groups that are housekeeping working groups dealing with aspects of communications, membership, networking, and outreach and training. I won't go into the details of the working groups now, but if you have questions afterwards, no problem. What are our activities? So the most visible part of, or our flagship to a certain degree is our congresses. This is the way that we started. When the initiative started, we thought, well, the idea is to create a hub where people working in core facilities can meet, and what is the best way to do that? To organize a wide congress. And so for the moment, we've had 3 congresses. So the first congress took place in 2014 in Institut Pasteur. Then there was a second congress in EMBL in Heidelberg. And the third congress last year took place in Belgium, in Ghent. And so the average participation of these congresses has been 300 people coming from, well, mainly Europe, as I said, but also congresses, people coming from further away. The next Congress, as some of you know, is going to happen in Portugal, in Lisboa. And so it's happening next year from 30th of June to 2nd of July in the Fundación Gulbenkian. And so it's the principal local organizing institution is the IGC, together with several other institutions. And so there is a website that I invite you to go and visit. And if you have any question, the address is simple. It's info@ctls2020.pt. So what is going to happen in that Congress? So there will be some keynote lectures. So we have 3 very different keynote speakers. So Florian Jug coming from Dresden to talk about aspects of machine learning and image analysis. Jan Husak, he is the chair of S3, of the European Roadmap for Infrastructures, and so he's going to talk about the perspectives at the level of research infrastructures in Europe. And Michela Bertero from Barcelona is going to focus on questions of open science initiatives and procedures. Then there will be several sessions on topics that are not specific to one field of specialty, but that aim to be hopefully of transversal interest. So on aspects of automation, of methodological and technological development in core facilities. Important that was stressed before is that however rich an institution and however big an institution cannot have everything in their own premises, which means that very often there have to be ideas about how to connect and how to share potentially resources. Beyond the plenary part of the Congress, there will be satellite meetings, and basically the idea is to provide slots to any community that wants to meet on a focused topic, and so that will have the possibility to have a room for free to discuss about any aspect of core facility life that they would like to. And so at the moment, the call for abstracts for the Congress is already open since October 15th, and the registration will open in just 2 weeks' time. And so I invite you to go and visit the website and see whether you are interested to attend, participate, submit abstracts, and there are many ways in which people from here, from i3S, can participate in the running this Congress and making it a success. So CTLS is not only congresses. As I say, this is the most visible part of our activity, but beyond congresses, we also organize and endorse smaller events because, of course, not everybody can go or afford to go to a big congress. So we have been co-organizing or endorsing many types of training events, more particularly on questions linked to soft skills and management aspects. So recent examples are that we were co-organizing with German bioimaging management course that takes place now every year in an island in the Lake of Constance in Germany. Very recently, there was a core facility management course in York in the UK that was co-organized with the Royal Microscopical Society. And so CTLS members could benefit both from reduced rates and bursaries to attend these courses. So in Institut Pasteur, where I work, so we are organizing at the end of November a course that is aimed for people who are technicians, engineers, scientists. So I would say more for people who are not core facility managers, and also for people who are, as we put here, who are currently not working in core facilities, but are wondering if this potentially could be an opportunity for them in the future. So at the moment, so this will be a 3-day course with lectures, roundtables, workshops, and shadowing visits. So people will spend 2 half days in 2 different core facilities of the Institut Pasteur. So for the moment, we have already closed registrations for the full workshop, but it's still possible to register for the first day of the training schools, in which there will be several roundtables and workshops. And hopefully, well, this is a pilot that we are doing. If the concept works, the idea is that potentially this concept of training school could move from one institution to the other, and so that like this it can be replicated with, of course, changes in many different institutions in Europe. So also, as I was telling you, we are putting in place a scheme of bursaries for a shadowing program that will allow people to perform short-term exchanges between core facilities, and our first call will be launched at the very beginning of 2020. What are other things that CTLS is involved with? We are a partner in a Horizon 2020 funded project that is called i3S. So i3S stands for Catalogue of Research Infrastructure Services. So this is a very ambitious project, I would say, that the idea is to create an open, trusted, and user-friendly portal, as is written here, that aims to list all kinds of research infrastructures in all fields of knowledge, so not only life sciences, but also hard sciences, social sciences, etc. And that basically the idea is that somebody who is looking for a certain expertise or looking for a certain methodology will be able, through this portal, to get access to one or several places that could be the place with which they could interact. So the project started just a few months ago, beginning of 2019. And so what is i3S? So the idea is that there should be a portal for all kinds of research infrastructure services. So research infrastructures being either at a local, regional, national, European level, so whatever their size and scope, and leading towards the possibility to have a kind of marketplace of research infrastructure services where people could locate, as I say, one or several options of places to go to and interact. And of course, for the project to be a success, it's very important that the core facility and research infrastructure community at all levels enlists and engages into this project. So soon a pilot version of the portal will be ready, and so we will be looking for beta users, for people who will be ready to test the portal, to tell us if this seems useful. So both people working in research infrastructures or core facilities and users, because it's very important to know if the users will resort to this portal rather than going to Google or going to some other kind of web-based resource. And so please, you can go and visit the website at the moment. So the domain is www.catris.eu, and we'll be communicating with you to call for potential users, beta test users. And so CTLS is an important stakeholder, I'm convinced, in the core facility world, but you should be aware, and we are aware, that we are not the only ones. So there are several other organizations in Europe and outside that are also active at this level. So in Europe, there is an organization called Core for Life, which is an alliance of currently 8 research institutions that try and bring together several aspects of their organization, one of them being core facilities. EU Life is another type of alliance, bringing together other research infrastructures, other research institutions. There is another one called Alliance for Life. Alliance for Life is based in Eastern Europe and tries and bring together institutions from many countries of Eastern Europe. And the other side of the Atlantic, I already mentioned it before, there is ABRF, which is the association, well, basically the equivalent of CTLS based in North America. And so to avoid duplication, because well, any association has, of course, very limited human resources and relies a lot on people being voluntary and giving time for the progress of the association. So it's really important for us to avoid duplication with all these other organizations and to carry out joint actions. And so I just want to show you one type of joint action that we are involved in is together with EU Life, we have launched what is called a small and medium-sized research infrastructure initiative, because at the level of the European Commission, I would say, is focusing, and this is relatively logical, on research infrastructures that are big transnational research infrastructures. So, for example, Eurobioimaging, etc., etc., and so, or EU OpenScreen that was mentioned before, or Elixir. So these are big consortia, disseminated consortia involving core facilities from many different countries. But so we are involved in active discussions with the European Commission and with other European organizations to recognize that the field of core facilities and research infrastructures is a real ecosystem, going from very local core facilities to transnational core facilities, and that you have to take into account all of this ecosystem. If you take one piece out, the whole building falls down. And so you cannot just look at the big infrastructures. And so this initiative was launched in May, and really the idea is to try and aim for a better recognition of all levels of the core facility ecosystem, and to try and enable a more sustainable funding scheme, notably for networking. Another initiative that I want to just mention very quickly is that Cost Action was recently submitted in September 2019, that is called Coliseo, which stands for Cores in Life Sciences in Europe. And so the idea is to enable, I would say, better networking between core facilities, especially oriented through Alliance for Life towards Eastern Europe, where we see very often that in research institutions and universities in Eastern Europe, core facilities are either non-existent or very, I would say, secluded. And well, hopefully we'll see if this initiative is successful. And so the coordinator is actually Nuno Moreno. That's certainly some of you know, in the IGC in Lisbon. And that's it. So if you want to know more, we have a website that I already pointed before, but here is again our website, a contact address. We are present in social media, both LinkedIn and Twitter. And CTLS publishes newsletters basically every 3 months. And so these can be downloaded from the website. And that's all I have to say. Thank you very much for your attention, and I open to any question that you might have. Thank you.
Thanks for the presentation. It was very interesting. I'm a researcher, but I have a question regarding your experience that you might have in certain core facilities. So sometimes they are, like in i3S, we have some core facilities that are very successful, but sometimes they can fall victim of their own success, meaning they have too many users. So what is the best way to move forward to avoid losing quality, or having people who are stressed because they are working too much, and because you mentioned that actually institutions should be the ones that should do the investment of maintaining the structure, right? Or should we get money from projects to try to improve the structure?
Yes, indeed. So there are two very different, I would say, situations. In some cases, it's a core facility gets very many users, both in its own institution and outside, but they are relatively, I would say, small users. So there, I think in our experience in Institut Pasteur and what I've seen in other institutions, the clue is training of people to become autonomous users. And I think this is, honestly, I don't think there is any instrumentation that you cannot say that you can train somebody. Sometimes it takes time to train somebody, but with willingness, a PhD student that really wants to have access to a technology will at some stage become an autonomous user and then be able to access by himself to an instrumentation, and so take some burden away from the core facility staff. And so I think a successful core facility that if they see that they become more and more successful should invest a lot of time in training so that they can have, I would say, a certain number of relays that can actually also then themselves train people from other labs. Then there is another situation is that we often see in core facilities is that there are 1 or 2 labs that are massive users of a core facility. And so, well, then a core facility, and we saw that this was the case in i3S, that a lab in that case must kind of replicate to a certain degree things that could be in a core facility, but that they need to have locally, because of course, a situation in which a lab is using 40 or 50% of the capacity of a core facility is not a healthy situation, because the core facility has to be open for people who are not knowledgeable in the field and must serve as guiding them to become maybe at some stage proficient. So I think these are the clues, working hand in hand with big users in the situation where there are big users, and so co-writing grants. So this is something that, For example, I do very often that we write research grant applications together with PIs to enable an instrumentation to come in the institute, but not necessarily in the core facilities. And so then it's important, so you investigators showed similar situations earlier. It's important to know that the instrumentation is potentially available for other users than the main person who actually wrote the grants, but not necessarily under the responsibility of the core facility. And so then the other solution is really training of users.
Thank you.
I have a question. If someone goes to a core facility, one of the questions is they want to know the price and they want to know the turnaround time. CTLS has something in place where a researcher can go there and see which core facility is cheaper or the turnaround time is short to go there?
No, CTLS per se, no. So this is one of the objectives of i3S, of the catalog that I was telling you about, that indeed there should be information about the capacities that are available in research infrastructures, about the procedures. So then, indeed, questions as what is the average response time, you could say, should be there, although sometimes it's quite difficult to quantify, because is the response time, is it the time between the moment that you send your request and that you get kind of first meeting with the core facility people to actually discuss what you want to do, or is it the response time between the initial requests and the moments that the experiments are actually performed? It's quite different because, as I say, very often the requests that a core facility gets are not specific requests that I want to access such type of instrumentation for such and such question. Very often the question is much vaguer. It's, I'm faced with such a problem. Do you think that you have a solution that can help me out of this problem? And so this means that very often there is a work of reformulation, meeting, thinking about what could be the different options. And then, of course, time takes longer because you need to really understand what is the specific question that the researcher has. And this is, honestly, this is, from what I see in many institutions, the case of the majority of the questions. Really the situation in which the question is very clearly and specifically formulated, it's a tiny minority of the requests that core facility gets. So these are the, I would say, the easy handling ones, but not necessarily the most interesting ones. The vaguer questions are often the ones that give rise to the most interesting projects. And of course, they take longer.
I'm a big user, so of the microphone. I have a question. So regarding, so of course we are talking about core facilities and technologies. So another partner, important partner, are companies, right? So I was wondering if CTLS has partnerships with specific companies, you know, to facilitate the use of the use of new technologies that arise and that can be, people can see how they work and if they are adequate for specific core facilities.
Yes, so CTLS really is I would say transdisciplinary organization. So there is not within CTLS partnerships with specific instrument developers in each field. That I would say there are networks, discipline-specific networks. So as I was saying, there are proteomics networks, there are genomics networks. So each of these discipline-specific networks involving core facilities and non-core facilities develop such kind of things. So CTLS per se tries and partner with two types of companies. So on the one side, many companies that develop softwares and solutions for management of core facilities, both at the level of administration and project management. And also another type of companies that CTLS involves or tries to involve for the moment with, I would say, mixed success. These companies that very often are academic spin-offs are basically core facilities working with a commercial or profit aimed organization, but basically they are doing the same job as academic core facilities. And it's important. So it was mentioned earlier that by one of the speakers that sometimes resorting to a contract research organization or to a company doesn't give rise to a very good results. And this is something that we are trying to overcome, is that really works with a commercial entity. And so this is something that we are trying to work on a little by little. Another aspect maybe of companies, because this is something that is quite generic, is the aspect of automation. So it's true that we are trying to work also with companies that are involved both in data generation automation and also in data analysis automation. And so CTLS is partnering with a society that some of you may know that is called SLAS. So it's the Society for Lab Automation Systems, because this is, I would say, an issue that concerns all core facilities, whatever their field of specialty.
Sorry.
Yeah, no problem.
No, just because something you mentioned before, the acknowledgement of the platforms, which I think is an important issue, and I think also here in i3S we all sometimes, lately we got messages, people should acknowledge the work done in the facilities. And I was wondering, so about this issue of authorship, because it's a hot topic in science in general, who should be an author, who should not be. So do you have any, what are your thoughts about when a platform technicians, managers should be authors of papers or not?
Well, I think that the general view on this is that when somebody contributes intellectually to a research work, then they should be co-authors. And so it's difficult to set a clear border as to when is somebody actually intellectually contributing to a work. So this is why I say that it's very important that there is a direct connection between the principal investigator and the core facility manager, because basically it comes from the discussion between these two people to decide, well, okay, what is going to be, who are going to be the authors of the paper, who and in what order these authors are going to be. And but at some stage, of course, after the results are produced and analyzed, it's very important that such a discussion happens. And as I say, unfortunately, what we see very often is that the core facility managers are not directly in contact with the principal investigators, but are only in contact with the PhDs or the postdocs. And this is not a healthy situation, because then basically it leaves the core facilities kind of with no way to actually influence the way that the papers are written. Of course, well, the counterpart of this is that the people from the core facilities must be ready to actually involve themselves in writing the papers. And so, of course, you cannot get the fruits of the work and be a co-author if you haven't actually participated in writing the paper and carrying out the work. But so it's a struggle, but honestly, I think things are progressing. Then at the level of acknowledgements, one of the things that we often see, and that I know here also is a problem, is that some labs consider that because they are paying for access or that they are paying for a service, why should we acknowledge? Because we are paying. And honestly, this is a stupid way of addressing things. However, how much you pay, it's only a contribution, I would say a relatively minor contribution to what a service or an access actually costs. If you start looking at what maintaining or running an instrument costs in terms of personnel, in terms of infrastructure, etc., you will see that the cost of what you are paying is ridiculous comparing to what it actually costs. So it's really just a kind of participation, and it's not because a lab is paying for an academic core facility that they should ignore their contribution in the acknowledgements part. I think really a core facility should be, if they have contributed to, if there is a result in a paper that is published, they should be systematically acknowledged. Then the only question comes at the level of should there be or not co-authors, and this is a one-to-one discussion between the corresponding author of the paper and the core facility management. Yeah, there's a question over there.
Are specific for core facilities in terms of funds that an institution can apply. For example, if I want to update some equipment in the core facility or to buy something new. So my question is specific projects that a platform or an institution can apply to have the opportunity to have funds for the equipment that we have in this platform.
Yeah, so this is of course a critical issue, and the problem is that the European institutions consider that this is something that should be handled at a national level, and that currently there are no European funds that allow to equip or specifically to equip, and this depends a lot from one country to the other. So as your director showed this morning, well, the situation in Portugal is that there hasn't been a call for this since 2001. There are countries that are, I would say, more fortunate at this level, but there are also countries that are in a worse situation than this. Now, very often, there are, on the contrary, calls at the level of Europe, such as the ERANET calls, or such also as the ERC calls, that allow to buy instrumentation. And although, you have ERC grantees here, and you know how constrained the situation is, how can you actually acquire an instrument that is going to be beneficial for an institution and not just for one project? It's constraining, but there are ways. Honestly, in Institut Pasteur, for example, there are several ERC grants that have allowed to buy instruments that are actually benefiting the whole institution and not just one single project. So, then at the level of CTLS, this is one of the aims of CTLS, to try and indeed map country by country what are the funding opportunities. But this is, of course, a very challenging task, because there are many countries in Europe, and honestly, the situation from one country to the other can be dramatically different. So I would say one of our objectives, of course, is not to have a kind of exhaustive mapping of the situation, but to use this as a lobbying means for countries that indeed don't have a very appropriate funding scheme for equipment, to say, well, look, we are aware that in such and such and such countries there are funding schemes like this and like this and like this, and would there be a possibility to actually try and upgrade what is available in one country to bring it closer to what is happening in another country? which means that indeed there needs to be knowledge of what is happening between the different countries. So hopefully CTLS can help with that. But it's honestly, it's a permanent struggle. One thing that you shouldn't ignore, because this also allows many core facilities to get equipped, is that we have a kind of, I would say, obsession in terms of institutional funding. But many core facilities get instruments through donations, and this is something that really shouldn't be ignored. There are people who are ready to sign checks for hundreds of thousands or sometimes millions of euros in order to have their name attached to a certain piece of instrumentation. Well, this is something, of course, that happens a lot in the United States because they are very accustomed to this kind of private funding of equipment. But in Europe, it exists, and I think it's really underused. When an institution wants to attract money like this from donators, equipment is I would say, relatively easy way to attract money, because indeed, when a donator gives money, they want to have visibility on what they are donating. And so I think this is something that should be taken into account in institutions like this one.
And in consequence, how we see the potential economical countries such as Russia or China or India, the possibility to have bridge
Mm-hmm.
CTLS? How is your overview of this?
Well, for the moment, as I say, we are really focusing on Europe because this is our core. And so what we hope is that in countries like the ones that you say, that's similar organizations will sprout. So in China, there are people who are moving to try and create a core facility organization, but the problem in China is that honestly the universities are in a very competitive situation, one against the other, and so when they start thinking about collaboration, it's a bit challenging. They spend more time competing against each other than thinking about collaborating. But hopefully the same way, so we are in a situation in which there is an organization in North America, ABRF, there is one in Europe, and hopefully there will be like these others in other continents, in other countries, and maybe then we can try and make a kind of, consortium of organizations that then can work together. But CTLS as such is not pretending to expand to other continents than Europe for the moment. Okay.
Any more questions? No? So I ask you to join us for lunch now, and we'll see you in the afternoon for the open live sessions if you registered for it. Patrick, thank you very much. It was very enlightening.
Thank you for the invitation. Okay.